I'm going to go ahead and just start sharing my screen so I'm ready to go. Stop. Oop, I always do that. Okay, am I in present mode? Cool.
All right. Yes.
All right, well, let's go ahead and get started. So I appreciate everyone's time today. We're gonna have sort of a relaxed, fun journey through some of the stuff that we've been putting together and talking a little bit about exploring data sets in the LungMAP data browser and in Terra, and talk a little bit about what Terra is in the browser are and what the motivations are around them. We're not gonna be able to cover everything. These are complex applications, but I'm hoping to give you a spirit of why they exist and how to use them a little bit. And if this is exciting for people, we can continue doing this. All right. So firstly, I'd like to thank Dave Rogers from Clever Canary and also Trevor Heathorn from the University of Santa Cruz for co-leading this demo today as we go forward. All right. So first, a little bit of motivation about Terra. Well, and just in general. So honestly, it's completely mind-boggling and fantastic, some of the projects we hear about nowadays, right? Like, so we started off with 1 human genome, and then you have the 1,000 Genomes project, which actually has 3,000 participants in it. And then, then you have 1 million genomes, for instance, in the All of Us research program. So this is fantastic. It's amazing the kind of data that we can get and the inclusivity that this points towards us being able to move towards, right? That being said, this is a lot of data, and it is very difficult for someone to leverage and manage and share and collaborate around at scale. And so while some solutions may have worked in the past, us moving into this golden age, this amazing age of computational biology, we have to rethink about how we're going to move forward in working with data as a group and doing big science. So in the past, the traditional approach has been for people to have their own local resources and to bring data to the researcher, right? And this works for even a lot of projects even nowadays. So this is still a useful thing. But fundamentally, when talking about doing large research and working together as in big science, this can be very problematic. Number one, it's not actually data sharing, it's data copying. And so you hope that you got the right copy that someone else is using, for instance. Because it's data copying, this actually is very expensive. Everyone has to have resources that will be able to size and manage this, be able to hold it. And I actually call this a metastic infrastructure. It's also, since everyone has their own unique flower and snowflake of infrastructure, the results are going to be different. It's very difficult to have actually reproducible results this way. And because we have to create infrastructure to the peak of processing, as opposed to the minimum of processing, it actually becomes very expensive because it's fixed compute resources. And then as far as security goes, since everyone's copying data, of course, everyone's using the data correctly, right? But the reality is it's not auditable. There's actually no security perimeter that you could actually understand. And so it's important for these data sets that we've been making to be able to move in a different way. And so the paradigm shift has been to move towards, in this case, a cloud-centric approach, but most importantly, bringing researchers to the data, right? So this is inverting the problem. But by inverting the problem, this actually fixes a lot of those concerns when you're talking about these large data sets or doing big science together. And so number one, it actually is real sharing. It's not copying. You use the same exact file that someone else is using. It's file sharing in place. As well, it is an opportunity to not only share data, but to share pipelines and workflows and such in a way that they are actually executable and actionable. They can be used immediately, already installed. Right. And so this does help with the reproducibility problem, as does containerization and other best practices that were becoming important in our modern day. This is cost effective because you only pay for the compute you use. You only pay for the storage you use, and it's not copying. So no longer metastic. Hopefully this is benign. All right. And lastly, since this is you bring researchers to the data is centralized, it's in one place. It allows you to have an actual security perimeter and allows you to actually have data security and be able to say it is secure. And so this is something that, as aspirational and a vision, but at least here at the Broad, when we started moving towards this, because our internal infrastructure, on-prem infrastructure, was not supporting the kinds of scale that we needed to do for our genomics platform and other large projects, the first thing you learn is that clouds are hard to use out of the box. Let me just be honest with you about it. And it's just because it's a new paradigm. It's very technical, all sorts of things. It's a learning curve. And so the reality is we started moving forward to create a solution for ourselves called Terra. But in making the solution for using cloud environments, we made it so it was an open solution. So not only can Broad people log into it, anyone in the world can, and it's positioned not to support our science, but to support global science. And this is something that we've partnered now with both Microsoft and Verily. It's currently leveraging, underneath it, it's leveraging commercial cloud, currently right now Google Cloud Platform, and we're working towards bringing Verily Cloud as well, Azure Online as well, to be multi-cloud. So it's very exciting to be able to talk to you a little bit about that today. The hope of Terra is that it is a hub within an ecosystem of many things, and it leverages, you'll see that it leverages a lot of different systems even outside of itself. And there's a big focus on it to be interoperable with other systems. And we'll see that today we're interoperable with the LungMAP DCC. And in fact, by Data Catalyst as well, which is important to us. And that's going to be really important to be able to leverage the kinds of resources that are available for consortia. As far as the kinds of things that happen in Terra, there are data libraries to be able to access both public data and managed access data if you have the authorization for those data sets. There are workspaces, which I'll show you, which bring together the concepts of security and data and computes all in one place. So you can work by yourself or with a group of friends or make something public. There are the ability to add in or search and find workflows that are cloud native and scalable. Same workflows are being used in other consortia, or it could be your own. You can bring your own labs in if you want to share with someone. How do I get someone else to run my pipeline? This is exactly it. They can literally run the same pipeline, the same system that you're doing. And of course, a solution wouldn't be complete without being able to interact with the data. And so there are some tools in there for interactivity. And you'll see that these tools are not tools that were made by us. They're tools that have been adopted by the community, like Galaxy and RStudio and Jupyter Notebook and those great things. And so once again, interoperability with other groups and systems. I'll show you these, but this is just a picture of showing a catalog of data sets. Pipelines, for instance, I can show you pipelines from the Human Cell Atlas Data Coordination Platform or the BICCN, for instance, the BRAIN Initiative. As well as other ones. And I had mentioned before interactivity and different kinds of environments that one can use that we work with. Also, this is a secure environment. We're not only FISMA moderate authorized, but we are FedRAMP authorized environment as well. This is very important to be able to work with the kinds of sensitive data that we have and keep our promise to our patients and our donors. So at this point, unless there's major questions, I'd love to be able to jump into our demo to keep us on time. So far we're on time, which is excellent team, and talk a little bit about Terra as a demo, because it's better to look at it actively. All right.
All right.
So as far as this goes, what I'll do is I'll do a little bit of an overview of Terra for about 10 minutes. Then we'll jump to our UCSC team, because I want to give a little context of what what the UCSC team is going to show us. We'll jump to them for a moment for the browser, and then I'll come back and work with that handoff a little bit of data. All right. So the first things first, let's go to Terra itself, terra.bio. I'm going to log in, and I'm using a Gmail account in this case. As well, I could, as long as it's in this case here, since the underlying cloud is Google, it would have to be something that works in that space, because otherwise you couldn't use the underlying resources. There's work towards that in the future, but for right now, anything that would work in that space would work here. And so this is Terra. And when we're looking here at Terra, the first thing to show people, and I'm not going to go through all the functionality details here. I just really give you a spirit, like a sense of what Terra is and how to use it. As people are interested, we can go into more details about things, but some of the high level landscape here. So firstly, under the hamburger, we call this the hamburger. You can see here that it knows me. I've logged in, and then at that point, it's going to be giving me all the authorizations and authentications that I currently have. So all the workspaces I should see, I'll be able to see and such like that. And under this, it allows me to have a profile, allows me to have groups that I will set up that I can share with, allows for billing to happen. Now, as far as Terra goes, Terra is an application sitting on top of the cloud. Do not charge people for using Terra. If you are a nonprofit or you're a charity group, you're not being charged for using Terra. That being said, when you're using Terra, you do leverage underlying cloud resources. So there are pass-through costs for using the cloud resources. We try to make you very aware of them as you're working through things. They end up being small, but it takes a moment to get used to that. But once again, we do not charge nonprofits for using Terra. So keep that in mind as you're seeing things. That being said, you'll need to have be able to interact with the underlying cloud. I'm going to skip over workspaces for just a moment. That is where the action happens. I'm going to move to our library. So in the library itself here, there's a couple places here. There's data. Data, for instance, is a place to find data sets that are made public, like large repositories. So an example of that might be interesting to this group is the Human Cell Atlas project. And here, for instance, if we click on this brings us to the Human Cell Atlas project, an excellent browser that happens to be made as well by Trevor and Dave. And so that similar handoffs that you're going to see today with LungMAP would also apply here in this browser. Just jumping back. And so some of these don't require authentication to access, like that one, for instance, is all open access data for now. And so it doesn't require that. And so you just go directly into it. Others might require authentication, and it takes care of that for you. It'll guide you through what you need to do next, either emailing someone or actually going through the application. But nonetheless, that's featured data sets. So there's also under here, our showcase. And this is a place to find workflows and tutorials and such. And so on the left here, you can see sort of a categorization of them. And you can also do a search as well. But at the end of the day, for instance, if I'm looking for Optimus, which is a pipeline, this allows me to find that. And then if I click here, it'll bring me to the workspace. And I'll show you that in just a moment. And then lastly as well, there are workflow highlights. So these are some highlights just from GATK, for instance, and some things that are highly used. That being said, there are lots of different workflows. You can go to DocStore to find workflows, as well as the Broad Methods repository, and then bring those back over into the system to use and attach it to your workflow. And you'll see an example of that. Lastly, and very much importantly, there is the support. And so we have lots of different support for using Terra, whether it's guides, videos, and tutorials, being able to request things directly, a forum itself, directly contacting someone, or looking at our programmer and developer release notes. So depending upon the kind of needs you have and what kind of person or role you have, whether you're a developer or you're using things, or you love videos like me, I like to see videos, I like reading text, they're available here for you from there. And of course, we highlight COVID-19 data and tools to make sure that those are in front and available to people. Now I'm going to quickly go into what a workspace is, because our browser team will be handing off data to a workspace. So I want to make sure you're aware of what that is. And then we're going to jump over to our browser team. And so in workspaces here, this lists all the different workspaces that are available to me, whether they're mine myself, or if they are new, or if they're featured, or if they're public, fully public. And I encourage anyone to just log in with a Gmail account and to look at these. And you don't have to set up a billing or anything like that. This is all just something you can look at and peruse and such. You'll know if you actually need a billing. This actual account that I've logged into actually doesn't have a billing account attached to it. Just to give a sense of a featured workspace, this is a pipeline that's used by the HCADCP. In this workspace, we see a description about what you're seeing in there. In this case, it's about the pipeline itself and what it's used for, what are its inputs and outputs, and the costs in the cloud to use it, giving different data sets so that you can get a sense of it and start playing with it a little bit. As far as data goes, this is a place to have both metadata. So this is an example of very minimal metadata, but metadata, and also data itself. In the bucket, this is the Google bucket attached to this for the workspace. So in this case here, the metadata is useful for running the pipeline, and the files are sort of runs that have already happened, and example data so you can do it yourself. This doesn't have a notebook. I'll show you a notebook in the future, but there's a place to have notebooks at, and then we can have workflows attached to the workspace itself, so you can run those workflows. Here are different versions of Optimus configured for either the version of chemistry or the species being used to use as an example. So if, for instance, you click on this, you would go to the workflow itself to start running things, and I'll show you that in a little bit. And then a work history of things that are running, or if they're done, or if you errored, or if you didn't, given your workflow running. Here, the team is really great. They've got them all green, so they all worked when they were running them. You'll see in my workspace, I wasn't so good at it. All right, but fundamentally, at the end of the day, that's the sense of the workspace here. It combines data and metadata, description, notebooks if you'd like, workflows if you'd like, your history to run things if you'd like, and actually has sharing and security as well, so that you know who it's shared with, and I can share with others if I want. I can lock it down so no one can affect it if I think I'm done and I don't want it messed with. And also, this is a wonderful cool thing here called clone. If I click on this and it's a featured workspace or a public workspace, I get a copy and I put it on my own billing object so that now I can run the pipeline, the whole thing. I get the data, the metadata, whatever it might be, with the pipeline already installed as they would have it in the workspace, and I can run it on my own dime, but I can run it and do whatever I want, bring my own data to it. It's not shared to the original people, it's mine, right? And it allows you to literally make a copy and have a living, already installed version of something to give to someone else, or if you don't want to share it with them. For instance, if you want them to run their own data on their dime, they could clone, but if you want them to work with you in the workspace, you could just share. So like lots of different options, and there's security around that, but that gives you just a general sense of this. All right, now I will pause for just a moment, but for time, unless there's questions, just a quick moment to see if there's any general questions. Okay, I'm going to then jump over to Dave, so we can talk a little bit about the UCSC browser, and then we'll come back to this and I'll go into more detail about things. Does that work for you, Dave?
Yeah, sure. I think I'll start to share my screen, and then Trevor can give us a brief intro. I think that's what we discussed. Right, Trevor?
Hi. Yes, so as Tim alluded to, the data browser you see here is a variation of the one that we used for HCA. We've made it a extensible plugin type of architecture so we can adapt for various uses, various platforms. It is based on data is stored in the Teradata repository using the metadata schema that was originally developed for the HCA. And that allows us to rapidly develop this variation of the browser. It provides a faceted search across all of the data sets that are available, and allows for us, a user to select a subset of data of interest for, like, example, an age range plus disease, and then use that subset of data and hand it off to, for example, to Terra for further processing by workflows in Terra. So I'll let Dave take you through the UI elements here and show you how it all works.
Okay, all right. Thanks for the intro, Trevor. Let's see, as Trevor discussed, then yeah, this was developed using it's a LungMAP branded instance of the software that was developed for the Human Cell Atlas. You can see that they're very similar, well, they're the exact same code set with just some branding differences. And but you can access this from the LungMAP portal. So if you go to resources and then data browser, then you're over here back to the LungMAP portal. You can see there's 3 ingested projects right now, but there's a few more, I believe, in the pipeline. And it's really great to see this thing full up. Now, some of the things that we share with that we get with this infrastructure is the ability to find files that are associated either within or across projects. A project is a high-level unit of organization in this browser, and it generally corresponds to a paper or a specific line of research, but generally it corresponds to a paper. So with these or study, maybe in dbGaP terms. So we have our list of projects then have the files that are associated with them, and we have metadata about the samples as well. But I'm getting ahead of myself in my demo here. So yes, we inherit the ability to slice the file sets, like I said, for a full project or slice across projects, and then we have metadata that's about those projects. It's represented in the metadata that metadata schema that was developed for the HCA DCP. Now, this is the schema is very kind of modular in design, and it's extensible and flexible, and it's meant to capture kind of the experimental graph of things that were done to create capture the experimental graph and as well as the analysis graph. So we have the structure of this schema. There's 5 basic entities. So we have the project that we just discussed, the files we get at the end, but we start with a biomaterial, and a biomaterial is start at the very top would be a subject, or it could be a cell line, or it could be an organoid. But the biomaterial is something that you start with, and then you do processes on the biomaterial to get further biomaterials, to further refine the biomaterial until you get to a library preparation. So these processes are instances of a protocol. The protocol is what I plan to do. The process is an instance of the execution of that protocol. So I take my biomaterials and I transform into biomaterials until I get to that magical transformation where I'm now taking a biomaterial and turning it into data. So that's our sequencing assay. And so now we have files. So we went from biomaterial to file. And then once we have file, then we have further analyses, further processes, which are algorithmic, that convert files into other sets of files. So this then is how the schema in the large sense represents the structure of the experimental data metadata, and then further on the analysis metadata. And so you can see this in the structure and read about it in the design sections here. There's also an overview section that talks about the types a little bit, and like we said, we have biomaterials. Here's the various types, the processes, the various types, and the protocols, and so on. And then we have our dictionary here, which then gives you all of the fields that are on each of the entities. And then, if you look at maybe the required fields only, since there's the set of required fields is much less than the amount of fields that we show here. And then a lot of these, a lot of the fields, a lot of the entities, the point of it is to have ontologized fields. So when we do have a field that's ontologized, it means it's got a set of allowed values that can be in fields that come from a specific ontology. You can come here and you can find out, it's like for cell cycle, we use the cell cycle ontology. It's used here in this cell line. We use links to the cell cycle that talks about how it was grown. And I know that my cell cycle ontology ID comes from, here's the graph restriction, means what are the allowed values. So we can link out to the actual ontology here. And so you can say, here are the allowed values for this term and more about what this term means. So when you're looking at the metadata or looking at the application and you want to know what a term means, you can look it up in our metadata dictionary and go into details about what this is supposed to mean. Sorry. I'll mention, let's see. All right. So we talked about within project and across project. And so if I wanted to go across project, I could say, well, I'm interested in species of mus musculus, but I want females and I want mouse prenatal. So this is a slice across all of the projects. It turns out there's only one project that has that kind of data, but these are the files, a slice of the files that are associated with just that, that match the metadata that I had selected. So that then here I export my selected data, and then I can request a curl command to download it. We always want to make sure people are sure about what species that we're using. So that's why we have this this box. If there were multiple species here, we'd force a selection, but there's only one, so we'll just select it for you. And then in the interest of time here, I'll just select this that I just want the CSVs. I can request my curl command. And then here I have it. I can copy the command. And then somewhere I have it. All right. Please go away. I have, I can paste my command here and execute it. And it's fairly easy to get the files downloaded to my local system. And here they are. But as Tim has mentioned, maybe our goal is not to download the files in our local system. Maybe this part is good for initial exploration, or is good for we can look at the metadata that's associated with the files. But what we're looking for is to analyze in Terra as part of this demonstration, and I'll get to that in just a second. So let me go back here. I'm going to clear my filters, and I'm going to talk now about project level access. So I see my three projects here. When I'm looking here at the project level, not really across projects, I can download the metadata for a project. And see here's a file that's got the metadata for this project, and the terms across the top are terms that you should be able to find in our in the metadata dictionary. And then, of course, we're interested in the matrices, and so here are the matrices for a specific quick access to the matrices that are available for a project. Again, just download here. And then if I look at a project detail, wanting to know more about a project, what I just selected this here, selecting this project gives me to this project project detail page. And then I can read about the description of the project, the publication that it's linked to, how to cite this project in this browser, and then also similar to these are the same functionality that was on the home on the top page, how to get to the metadata for this project, how to get the matrices for this project, and how to download the files. And here's the actual downloading. You're able to download the directly download. So I can also, rather than going through the CSV flow, I can directly download. Oh, this is the curl command. I was expecting a direct download here. For these files. Okay, so, but where we're trying to get to is this export to Terra flow here. And so I can...
Dave, before you do that, what's that analysis portals thing at the top?
Analysis portals. Oh yeah, thank you so much, Tim. Yeah, so here we have, this project is linked, is referenced in Cell by Gene. And that gets you to the Cell by Gene. Here's the Cell by Gene Explorer, and the other one was the Cell by Gene collections list. So these projects are linked out to Cell by Gene, and it's also linked out to, it's also linked out to the LungMAP apps. So we have this, the Shiny Cell app here for this particular project.
Thank you.
Yeah, yeah. So this is using some of the infrastructure developed to link out to the various applications. We'd love to see this linking out to the cells.ucsc.edu or the Broad's single cell portal as well. Right. So if we're going to go then get this project over into Terra, I'm going to just select and send the matrix files here. And then, whoops, here we go. I have a defect here. This is a request curl command here. But let me do this a different way. So I can also select this single project here, export the selected data there, and then I can select Analyze in Terra here, and confirm that the species I wanted, I'm going to select the H5s, and I'm going to get a link to export in Terra. And so this has sent me now over to Terra, and it's saying I can start with a new workspace, or I can go with an existing workspace. I happen to have a billing account set up, so I'm going to go with a new workspace, and I'm going to call it LungMAP Demo 2, and I'm going to select my billing project, which is this another Anvil won't mind. It's Clever Canary Anvil Dev, and then I'm going to create my workspace. So this is basically me selecting the matrix files from the project that I was interested in, coming over to Terra and creating a workspace. And now you can see that it's importing. It says data import in progress. It's got this data import in progress over here. This takes a couple of minutes to go. Tim, I don't know if you want to fill in here or describe what's going on.
Yeah, I can jump from here if you'd like.
Yeah, sure.
Okay, cool. And so would it be okay if I share my screen and start working from there?
Yeah, sure.
There you go. It's done.
Here it is. It's done. And so then I can say, you saw what Tim, refresh. It's asking me to refresh. And then, okay, so here I have, and these are things we should recognize from a metadata schema, analysis protocol, cell suspensions, and so on. I want to say, let's look at the donors. So here's the donor information, biological sex, any diseases they had, information that's wrangled about the donors, and then the files themselves. Here's the files that I exported. So, and here's the URIs to the files right here, the DIRS URIs. So I should be set up to then in Terra create a notebook to analyze these files, or use a workflows to analyze these files. Yeah. So there you go. I think that's it for us, Tim.
Thank you. Are there any questions for Dave before we move forward?
Yeah, I can just add that we're in the process of migrating this guides section into LungMAP that has a little bit more information about how to use the browser and how to accomplish some of the use cases that I just showed today. So look for that coming up soon. For now, you should be able to map whatever's in these guides section directly to using LungMAP.
Awesome. Well, thank you so much, Dave.
Okay.
All right, I'll jump over to sharing now.
All right, stop share.
Hopefully everyone can now see a Terra workspace again. All right. And so there's different ways of getting data into Terra. One way is to literally bring data yourself and upload it using typical commands in the cloud like gsutil to upload to the underlying bucket that's here. Another way is to go into the data, once again, the data libraries, and in our case here, LungMAP, and to hand off data or bring data into the system in that way. And so what you saw for the LungMAP was handing off metadata. And so let's see here, I did the same handoff with the same data set in this workspace here. And so what you saw, it was handing off metadata, but not the actual files. Instead, it's handing off links to the files. And so what that's doing is trying to make sure that we are not copying things left and right and making people pay for storage left and right for these kinds of resources, but instead are being very efficient with the use of it. And so, for instance, the storage for the LungMAP files for this are in our data center. And so those files that are handed off are actually just there and stored there and paid for the storage there. So one's not paying for storage for that. Once you do analyses and anything that you do in your workspace that's stored in your workspace and that's your own and that's under your control and under your billing and those kinds of things. But in that case, the metadata was brought over, as you saw, using these tables here. And you can do all sorts of things with the table as far as making new columns and being able to select or unselect columns and then save those selections or ordering. You can move things around by move like this, and saving those as views so that you can really customize this to however you want to see it. That being said, you can also just download this metadata and use it however you want to use it. We're not locking you in here, all right? But nonetheless, these are really useful because we use them, as Dave mentioned, to programmatically interact with and to leverage in like analysis, for instance, because this is a great data table to be able to do analysis in Notebook, for instance. Or for instance, if I set this up the right way, I can set it up so each of the rows is a different run, and then I can run a whole bunch of data through a workflow. And that's actually how we do it when we're running thousands of workflows over data sets. We literally use these data tables and we make sure they're all lined up, and then we say run them all, right? So it works in a lot of ways. But to go back here for just a moment, for this workspace, I am currently using 19 cents a month. So just to give you a sense of the cloud, we always try to give you a sense of that as you're going along. Now for this workspace, we'll jump back just again. We are using the Morse data set. This is not in any way an attempt to analyze the Morse data set. If you want to look at the analysis for that data set, you should go to the LungMAP browser and go to their publication and see what they did there. This is using a very small fraction of that data set to be able to show you functionality, and that is it. We are not the experts on this data set. Ed knows his stuff. I don't know Ed's stuff. Talk to Ed. All right. That being said, we are looking forward to doing more of this and being able to partner with people to be able to showcase work in this kind of spirit. But please, if you are interested in this data set, please talk to that great team. Now, for instance, I came to this workspace and I want to run a pipeline, right? And then looking at the workflows, I know, okay, so my pipeline, I'm going to run, in this case, the Optimus pipeline. It doesn't have to be, you could upload your own pipeline. I would love to see your pipelines up here and be able to see what you're doing with it and sharing amongst each other. Like, I really just want people to share. But the reality is if I want to use Optimus, for instance, this is a row for every file, but I kind of need a row for my file triplets. I need my read one and read two and my index all together so I can run those as inputs for each of the pipelines. And so you could simply just download this and then edit it in Excel, or just upload from Excel something if you'd like a TSV file, if that's what you're comfortable with. But I wanted to show, for instance, that you can use notebooks to interact with the data table. And so to use a notebook, one has to spin up the cloud, like a notebook environment on the cloud, right? And so because currently we have storage. And this is an example of how to create a cloud environment to be able to work in interactive spaces, whether it's a notebook or whether it's Galaxy or our Studio or an RShiny app, whatever that might be. We support a lot of different applications, but you'll need something underneath it to run it, right? And to store things. And so this is an example. So in this case here, this is a default that I could create, and this is giving you a sense of what kinds of cloud resources it's using. None of that's going to the broad, that's going to the underlying cloud, and those are just pass-through costs. It just gives you a sense so you know what you're doing as you're going. I could also customize this. And this, although what I was going to create was something that would have Python and R environments in it, as well as some GATK tools as well, you could do other things as well. There's lots of different environments and versions that one might use, including having custom environments. And if one is interested to know what's in this environment, if you click there, you can see the different packages installed for Python, for R, and if there are additional things like, in this case, tools, but there might be like the C libraries or whatever it might be. But this gives you a good sense of what's inside of it. So in this case here, the defaults that I would have ran include, for instance, GPU, being able to control how many CPUs you're leveraging, how much memory you're leveraging, if you're using GPUs. If you have any startup scripts you want to add into that VM that's being spun up, you can talk about the different kinds of VMs if you'd like to. Auto pause is really cool. If you are running an analysis, or like if you're working in a workspace and then someone's like, hey, come over here for a minute, and you run off, it'll pause it for you after this amount of time so that you're not burning up any computes or anything. And then the persistence disk is just its unique disk that's being stored and being used for that. And so if I, for instance, back here, if I had just hit create, it would create one of those instances. I'll create it just to let it run. It takes a couple minutes to bring it up. It creates a VM. It creates the persistent disk. It maps that persistent disk to it. It takes the container that you selected. It brings it to the VM so that those software are available. It takes a couple of minutes. But I'm going to move forward the presentation, but I just wanted to give you a sense of that. So once that is up, it'll say it's running. And in that case, then you can actually edit notebooks and run notebooks, but you can still see notebooks without doing that. So we're going to use that to move forward for the demo. And so there's a couple notebooks here. Actually, let's see here. Let's start with one. I numbered them for myself and didn't actually use it. So let's do number one. So number one is allowing me to update that data table with a new entity. And so if we look back here, this lane file entity that's very, very cleverly named, it is not in the original thing that David had passed over. Files was, and I want to restructure this. And so once again, I could download that, but if you work with programmatic things, or if it was, for instance, if you wanted to copy this workspace, clone it, and then do your own import, the same metadata schema will be used, and so the same notebook should be useful for it. So it's a way of a little automating things a little bit. And so in this case here, what this is doing is this is interacting directly with the data table. And in this case here, I'm a Pythonista, so I am programming some Python to be able to pull in, to read in that data table, to be able to then, in this case here, restructure it a little bit. And then after restructuring it, I'm checking the new thing that I'm making, that lane file data table, and then to send it back out to the data table. And so this is just uploading, managing, reshaping it, and sending it back out. But what that allows me to do, it's very logistical, but it allows me to sort of automate what I'm doing and be able to do this, especially if it was very large, be able to do it very easily for myself. And what that ends up giving me at the end of the day is a new metadata table here, where it has the lane and file information, but it has, here we go, the index, the read one, the read two all together. So for each row, that's a run of a workflow. And that's going to help me run multiple workflows if I wanted to. Now, in this case here, it's just a fraction of a lane. And so there's other things that I can do to actually run the full lane together. But as a demo, I'm just going to run a small piece of data. You'll notice also as well here that there is one row that has a whole bunch of other things in it that others don't. And that's because I already ran the workflow once and it made outputs. And when it makes outputs, it says, okay, I ran this row. These were the files associated with it. Okay, new files are being made. I'm going to make new entries in the column and I'm going to update those for you. So if you want to know what BAM or CVS file or whatever it might be went with these files, this is a record for you. And you could download it and use it as provenance, or you can literally use this in the notebooks that you use for analysis. So it sort of keeps that for you as you're going. Now, that's great. So I've made this stuff here. How does it look like to run a workflow? All right. And once again, a lot of this is a Martha Stewart cooking show, but just to give you a sense of how things, hopefully this is still giving you a sense of how things work. And so for workflows, as far as workflows go, I have one already added here, but to give you a quick sense of what it looks like when people are adding things. If you click on workflow here, it allows you to go to either the Broad Methods repository or DocStore. I always recommend, both are great, but I recommend DocStore. That's a great community resource for people to use. And then it's got here 1,094 workflows that could be imported into Terra or in other places to use. And so if I search for Optimus, there's many different pieces of it and the actual thing itself. And then I know that, for instance, like Warp is our actual workflow repository. So this is right. This is correct. And for instance, if I click here, for instance, this is the pipeline itself. If I click here, it'll go and you'll see a very similar thing to what you saw with the Dave, where it asks you where to put it. And you can select your workflow and your environment. So for instance, see here, I could go to the overview. That's where I was at. If I hit import, then it'll import it. But I've already done that, so I don't want to do it twice. So let's go back to Terra. But that's how I brought it in. All right. So I want to use Optimus. I clicked on Optimus here. and this could be your pipeline. This could be what you bring to the group in the community, right? And so this is telling me this links out to the GitHub repo, so you can see it, or you could just look at the WDL script right here if you'd like. And you can actually download that if you wanted to, but I do recommend you going to the GitHub repo instead. And what this shows is the ability to have, for instance, inputs, because pipelines need your inputs, files that are going in, and then also outputs, so files that are going out. And these are the files that are collected in that data table. So like you saw BAM column, that's what it was doing. It was saying, okay, this output BAM, what do you want to call it? I'm gonna call it BAM because I'm not creative. And so it makes that column and sticks the link there. Now, the files are in your bucket anyway, it just helps you find it. But going for inputs here, there's a couple inputs that are required, and then there's also ones that are optional. Now, there's a lot of inputs here, but that's true with every pipeline. You get to know your pipeline, you read the documentation for the pipeline. But for this example, I'm going to do something very minimal. The GTF is needed, and so I have a link to a GTF that I'm using that actually, if you clone the Optimus workspace, it comes with it, so you have it, as well as reference FastQ and FastFile, and then also the star reference. And so these things are available once again, if you clone the workflow itself, or you can make these if you want to work on a new reference. And then I'm going to have lane file ID 1, and then this FBB read 1 and then also read 2. Those were the columns, the read 1 column, the read 2 column, and the ID column, right? To be able to, that are in my data table. And so what this is saying is for every row that I select, go to those columns and plug those in for the runs. It sounds a little complicated, but the reality is you always have to put your inputs into your pipelines. The question is, do you want to do it once, or do you want to do it 1,000 times? This allows you to do it 1,000 times if you'd like. For today, I'm not running 1,000 things. Instead, what I'm going to do is just select a data set here. And so for instance, this is the one that I'd done before in the past. I'll just select this one here. Once again, PFB index, PFB read 1. These are the columns that it's leveraging. I'm going to select another one, hit OK. And so now it knows I'm just running run row, and this is where it's going to get that data, and I hit run analysis. I've never run that one before, so we'll see what happens. But the whole point is that's how it feels like to run these things. All right. And so that's now being queued and stored to being able to be ran. And then here, let's see here. I have already ran some stuff. So let's see if it's there. Yep. And so this is an example of the job history. Green is good. It means it's finished and it completed successfully. Red's good too. It means you're learning, right? But it has not completed successfully, so you still have to rerun it again. And so I failed to run it once, and then I fixed it. And this helped me understand what was my error, for instance, when I went into a little deeper, and it allows you to go in and, for instance, go and see the files themselves to work with them, or actually go into the submission and look, and this is the error. And that helped me figure out that I had put the wrong thing in the input, for instance. But you can also get to logs and all those kinds of things, and timing charts, for instance, for the job history. Blue means it's running. And so that is running. And so, but that gives you a sense of running pipelines in this. But in the spirit of a great baking show, I'm going to move forward and grab something that's pre-cooked. All right. So workflows. So we talked about workflows, and this is now what it looks like with the environment running, for instance. So now the notebook. Say, for instance, I've already magically created, ran data. So we got great data from the LungMAP. The DCC team worked really hard with the LungMAP scientists to get that into the DCC. And then that was handed off to a cloud environment because there's resources here that might be exciting for you. Also note, eventually, once we're finished with integration, this is going to be integrated with BDCAT as well. So although we're looking at open source data here, openly managed data here, the managed data should work the same way, which is a real boon, I believe. But you've got that, you ran a pipeline on it, and now you want to do analyses. Now, fundamentally, if you wanted to, you could download the Loom file if you wanted to and work on your local resources. No one's stopping you from that kind of thing. But if you wanted to stay in an environment where you're working with a large group of people, you want to collaborate around a notebook, for instance, or a Shiny app that you make available for them, then notebooks become, I think, even more helpful, especially with big science. Or if you're just trying to process something that requires big memory, big resources, GPUs and stuff like that, literally making those resources is just clicking on a couple buttons and then hitting run, right? So there are a couple things that one could do, for instance, and I'm just going to very briefly, given time, show that notebooks work, basically. But for instance, the Optimus output creates a Loom file, and that needs to be converted to a file format that Seurat more understands. And so this is an example, for instance, of installing packages. In this case, we're using now using R for these, but installing packages that are useful for that. And then after that, copying it over using SCEasy to convert the file, and then being able to send it back to my bucket, right? So very logistical, very easy, but it's super easy when you have a notebook that just helps you do it. And you just change the file name, right? That you need to, where's it at? Change the file name that you're using to copy and work with, right? So more interesting, another example, not super interesting. Honestly, if you want to look at the analysis, you should go to the paper. But another example is after you do have that file that Seurat could use, working with it in the notebook, if you feel like that makes sense for you. And so this is an example just showing you that that's possible. And so this is, once again, this is an R, this is installing a couple things to get started, reading in the RDS file that I made in the previous notebook. And then this is going through the standard Seurat tutorial. So a lot of this stuff, if you've seen Seurat, should be make sense to you, seeing the UMAPs, selecting variable genes, being able to do a little bit of differential expression off the clusters based off of just like the standard stuff they have there. And none of this information is super important. You should once again go to the paper to see genes and stuff like that. But the point is you can do these kinds of things. And this is just using a subset of the data, but depending upon how big the data is with larger resources, you can do even more. But that's an example of running through a pretty standard tutorial and what it would look like for the notebook, because you have to copy the data in and copy it back out, and it goes, make sure you see that kind of thing. And for instance, if I wanted to share this with a good friend, maybe I'm going to share this with Jeff. He's a good friend. If he, for instance, went through and cloned this, he would have all these notebooks. He would have the pipeline already attached. He would have all the data linked, right? And then the new things that he makes would be his own. And he can run pipelines as much as he wants or not wants, depending upon his appetite to do that in the cloud, right? And he would be responsible for that. I wouldn't be worried about it, but I could give him the resources to do that. And as well, if he wanted to, he could go into the notebooks itself, and he could edit it and play with the actual notebook. And that's one of the real, I think the powerful things of this is that you can share things that work and are already installed and running and modify. So if I had this and I wanted to change the actual file itself that it was running on, but do the same commands afterwards, like I actually did to make this. I had an example template before this and then added this in and then went through and ran the different cells and did this in about 5 minutes, which made it really easy for me, right, to get onboarded and to get a sense of the data set. So hopefully that inspires you to do greater things than I did in a notebook. And so just dropping back for a moment, I'm going to do our last slide of the presentation, and then we might have a couple moments for questions if there are questions. And so now I think it's a good time to ask, so what, right? And so it isn't, what does this mean for you, right? Now, this movement towards making these kinds of resources is really important for the democratization of genomics. Right now, we're in a world where kings and queens do analyses, and many people don't even have the infrastructure, and we're changing that. This is to support global science to be done together, and we are changing that, right? And that's amazing. And we know that through COVID-19, for instance, with the pandemic, we know that we're all humans, so we all have this, we're all the same biology for the most part, right? So all of us working together to understand that, our human health is very important, especially when it may affect one population, but go to others. It's a team effort. That being said, you might have resources already. So in that case, so what? Well, there are things that might be exciting for you. Number one, just individually, uploading your own pipeline to run at scale for cloud resources. If you need more scale than you currently have, this is something that can be used for search and to be able to get those big projects done without having to pay for servers that will be idle otherwise. This is a cool way of making paper resources so that you can in your paper say, hey, with this link, you log into Terra, you can clone everything, and all my notebooks are live, active with pipeline attached. There's no better way of having a reproducible environment. This is it, like with the bow on top of it, right? It requires work to get there. I believe it's valuable. It may depend upon your paper, but it's an option for you. You can work with people in a shared space, in a private space, without having to worry about whose infrastructure or institutional this or that. It's a very easy space to come together, right? Even in private with just a collaborator. I don't even have to know. You just log in and do it yourself. Accessing data from other projects. HCA is one that you might be excited about it. There are other ones as well. So, going in and being able to work with the data is really cool. If there are some NIH data sets that we have as well that require managed access, it's pretty easy to update your profile from any of them so that you can just get access to them. So that's something to think about. And then leveraging pipelines from other consortia, like you saw me do today, where that pipeline started with the HCDCP. It's now being used in the BICCN, and it may be used in this consortia depending upon agreement. But that being said, it was really easy for me to grab it and use it, right? And thinking of other spaces outside of ourselves, thinking about big science. This is a great place to do consistent processing. It doesn't have to be my pipelines, it could be your pipelines. I don't honestly care. But a point of having consistent processing done and being able to share, especially for assays that are forming, right? Being able to help people get to new assays, onboarding them to it, or assays, honestly, that are neglected a lot of the time. So we don't see those pipelines available to people as much, but they're just as important readouts. So once again, helping other labs ramp up on new assays. Performing joint analysis together. As this bright group of people think about things to do together, you'll need a place to do it together at. And in those situations, consider this. You don't have to use it, but it's available to you. And then integrated and target analysis across consortia projects. It's a big thing working between consortia, but it's a great place to be able to actually have the data in place to do that, depending upon the consortia and the power to do it. So this opens up opportunities, but of course that is still work ahead of you. But those are some ways to be creative with this. So I'd like to stop for just a moment. And unfortunately, we only have about 1 minute. But if there's a question, I'd love to answer that.
Hi, Tim. There is a question on the chat. Jeremy is asking, can you accommodate our notebooks apart from Jupyter?
Yeah, so great question. The second and third notebooks that I showed were using the R language in a Jupyter notebook. That being said, you can also use RStudio if you would like. You don't have to use the Jupyter environment for that. And a lot of that sort of like what's installed in the VM itself comes down to the container that you're using. And there is ability to upload custom containers potentially as well. So there's a good amount of flexibility there. Cool. Thank you, Jeremy. All right. Well, we're available. Feel free to ask us questions. Oh, go ahead.
Yeah, just a comment, Tim.
Yeah.
As we started this whole project, seeing that we were going to work towards a pipeline that would be able to be accessible and usable by the entire community was just a great hope. And I see it in front of me, so it's really exciting to see it. I can't use it, it's too complicated for me, but I see its power. Training people, so we're onboarding, we have 4 or 5 folks in Jan's group doing computational sciences, and we're onboarding new people. We have our own pipelines, and it's what we're used to, and we train that way. And I guess the issue is training. When we bring a new computer science or epidemiologist, biostatistician in, what kind of training and how long will it take? And the troubleshooting at a distance may be harder than doing it in our own shop where we know our own troubles in our more modest pipeline. Thoughts?
Well, firstly, I'd love to see your modest pipelines in Terra because I would imagine they're actually not modest and they're very useful. That being said, as far as migrating to another infrastructure, there is always an onboarding, and that's why we put so much effort into the actual support itself, the videos and all those kinds of things. We also do have events to help onboard and train people. So if you do actively have people that you're interested in teaching, some of them are going to just hit it running with just the videos that we have in the blogs and the forums, and we have a help desk with the email and those kinds of things. So some will hit it running just by going through some of the videos. Some may need more, but there are opportunities, and just let's talk. Yeah. And Jan, I'd love to see your pipelines in here, so it'd be great to see that. So
train them, just don't take them.
Huh?
They're so valuable. These folks, getting them trained takes a long time.
And my understanding is that, so each center may have their, creative pipelines for the, research purpose. But for NIH, eventually, hopefully you had to, using a common machinery. So come out to the thing like the fits the fair standard. So this is super important. So my question is, if let's say if you said that you have like a video, something for if a new investigator analyst coming, we can ask them to since they're gonna to learn pipeline anyway, maybe we can have them to learn this and just register account, something like that. And
yeah. Right. So
how to do that? Yeah, basically.
Right. So as far as onboarding someone onto Terra, there are videos I can send you those. We have a YouTube video. I can send you the YouTube with all the videos in it. And then we have lots of blogs that describe like how to do like account setup if you want to do that. So that's all, we're very well documented, and we have a help desk. So if there's a question, you can just get the email and go and ask them questions. So there should be support for you, and I'll send those to you. And if not, if that doesn't work, just email me and we'll keep pushing on that.
Sure.
Yeah.
So it's a free registration, right?
Yeah, you can register, and the only time that you're charged for something is when you use the underlying cloud. So when you're running a pipeline or running a notebook or storing something, you can go through and you can work with it. You can even look at the notebooks and stuff like that, and like, in other places just fine. Even so, and once again, we're not charging you for that. These are pass-through costs. It just and they end up being, as you saw, pretty minimal unless you do something big. But nonetheless, it is the reality of cloud computing. So
that's great. Thank you.
Yep. Cool. All right. I know we got to jump to another meeting, but I will send you an email, Jan, and copy Jeff. And thank you, everyone. Thank you, Dave, and thank you, Trevor.
Bye.
Thank you, Tim. Thanks.
Bye,
Lana.
thanks.
Thank you.
Wonderful.